This session is on scale and harmony in the brain, and I'm pleased to introduce Paul Verschure, who is from the Catalan Institute for Bioengineering in Barcelona and the SPECS Lab, who will speak on the multi-scale temporal codes of the mixed parallel serial virtualization memory of consciousness, evidence from human intracranial recordings across perceptual, cognitive, and behavioral tasks.
That was the abstract.
That was the whole abstract. Let's welcome Paul Verschure.
All right, thank you very much for the invitation. It's great to be here. So, yeah, the title got a bit out of hand, but I couldn't make up my mind, and I hope in the end you will all agree it made sense. So, as several among you, I have a theory about consciousness. And I would like to investigate that theory now, looking first at, let's say, some theoretical considerations that we have elaborated using different kinds of robot systems. And then we're going to look at some work we're doing right now with human intracranial patients. However, I also have I use a definition of consciousness, and I have a declared statement on its function that I will illustrate to you. Okay? So, consciousness pertains, in my proposal, to the maintenance of a memory, a transient memory system, that has virtualized the state space in which the agent operates, that is unitary and sequential, and delayed because it's a form of a monitoring system, if you want. And I will explain to you what I mean with that. And the function of that system is to actually extract norms from the environment that can be applied to the optimization of parallel real-time control. Okay, so let's hope that at the end of the line, this gets clear. Yeah? So, why did I come up with this hypothesis?
How are you? Do you want to play music?
Well, we're doing a lot of experiments with human-robot interaction. Okay, so here we have an ICAP robot built by Italian Institute of Technology, interacting with Vicky Veluzzi, one of the PhD students, and they're engaging in the game. Okay? And this goes on for a long time, and the robot generates dialogues, and it generates social expressions, facial expressions, gestures, posture, and so on, to maintain this dyadic interaction. Okay? And what you discover doing this kind of work is to make these interactions effective. The agent, the robot itself, needs a very elaborate model of the other. But now to build this model of the other, there's very little to measure. Because the robot cannot know whether Vicky is bored doing this, whether he feels forced to do it, and so on. So this model of the other in the dyadic interaction is fully virtual. It's only based on inference, on hypothesis of the other. Okay? So this is one step in this notion of virtualization and optimization. But we can go a step further.
I will pull the octopus to the back.
All right, so this robot moved the octopus, this reddish object, towards itself. And now it has the octopus. Okay? So this was part of a broader experiment where we were addressing the question, how can an artificial agent actually acquire a language, or robotese? How can the robot acquire the ability to communicate with humans using a language-like and also grammatically comprehensible expressions? And behind that was an elaborate autobiographical memory we built. And in a dialogue with the robot, you can actually inquire or inspect this autobiographical memory. And you might have heard earlier that the European Commission is adopting laws that AI systems must be self-explanatory. Well, this is the first AI system that actually learns about the world and also learns to explain itself to other humans. But let's see how comprehensible it is.
Why do you have the octopus?
Why? I asked the octopus because I take octopus.
Okay, so what's going on here? Sok is asking the robot, why did you take the octopus?
I take the octopus octopus because I want octopus.
Okay, so the robot says, I take the octopus because I want octopus. But you saw the interaction earlier, where it was actually asked to move this object toward itself. And that dialogue and that interaction has been compressed now in this notion of wanting to achieve this goal. And that is based on the notion of that the control architecture that's behind this robot predicates the whole cognitive structure that it builds on an elaborate set of processes that just deal with states of the self. So the whole cognitive machinery is predicated on a deep grounding in the physical existence of this machine. But now let's go a little bit further in what this kind of modeling of the other really means. So here we have like a recursive robot because we have a robot that controls another robot that's learning to balance. So this robot will be positioned. There will be a cue. It's like a conditioning experiment. A weight will come down at a controlled speed, and the robot will have to learn to balance against this predictable perturbation. And what we have shown, and this is a very new way to think about motor control, is that in the standard ideas of motor control, first you think about the feedback controllers you need to maintain your balance against such a perturbation. And then second, you think about how you can anticipate the impact given the perturbation that you receive. And with that, you can now optimize your feedback controller. But if there's a signal, let's say a sound, that anticipates the occurrence of this impact, you can start to build a model of the world that says, well, when this event happens in the world, I can predict I will receive an impact. So you start to build a forward model that helps you to predict states of the world, and you use that to control your feedback controllers that maintain your balance. But what we showed is that this system can work very efficiently by not necessarily replacing the feedback controller at this primitive level, and that's the standard models that people study in motor control actually propose that. It's the famous Kawato model, where you would say, well, a feedback controller is fast, but it's imprecise. So I learn to predict, to replace this primitive feedback controller. What we're saying here, however, is that the feedback controller is now receiving new error signals that, if you want, are counterfactual. The forward models are basically saying, well, this event will happen in the world, and given that this event will happen, if I just adjust the error signal in the following way and shift it in time, I can now capitalize on the capabilities of the feedback controller to react properly. And what we've also shown is that this first stage of the process maps rather well on basic structures in the cerebellum, and this cascade of forward models on which you can build it relates strongly to the dense interactions that exist between the cerebellum and more frontal cortical structures. Why do I feel so confident about that? Well, we made a distinct prediction with this model, that is that the basic control loops at the cerebellar level would already represent aspects of the plant, that what they control. And a very recent study has shown that to be the case, that the distinct temporal tuning of these control loops in the cerebellum correspond very directly on the details of the biomechanics and the controllability of the plant. Like, to send a signal to your toes will take a bit longer than to send it to your eyelid, for instance, right? And so these internal models reflect that. So we have some direct anatomical and physiological evidence for that contention. However, the cerebellum in the human contains about 15 million of these control loops. And they're all segregated. That means they don't talk to each other inside this structure. And they're highly relevant for the real-time control of our bodies. We actually were the first people, the first group to build a neuroprosthetic system that was round-tripping signals with the brain. So it would read from the pontine nuclei, process the signal, and send back a stimulation signal into the deep nuclei of the cerebellum. But if I would just compress that system on a chip, and I would like to replicate the cerebellar loop, I would have a stack of chips of 150 kilometers. So it gives you a sense of the scale of this control system. It's all compressed in your skull, right? So what's the big problem with this? How do I now optimize all these parallel controllers, right? So I have my feedback controllers. I just showed you one for posture, but I have 15 million more. And at this level, they don't communicate with each other. And they get information over cascades of predictive models that don't know really what's out there in the world, but given the experience, they have predictions, they have hypotheses of what the world could look like. So to make that work reliably, what you really need is an additional controller, if you want, that is now sequentializing the operation of your parallel real-time control. It has to unify it, because now you want to extract a single objective that you say, well, did I do it right or did I do it wrong? For me now to say, did I do it right or did I do it wrong, there are millions of these circuits actually active. And some will be doing stupid stuff that you might want to suppress, right? So in order to capitalize on this kind of predictive real-time control, you need to think about a second level of control that is fully virtual. It's virtualizing forward models that in themselves are virtual. It is unitary, because at the end you can control only one plant, and it's sequential. It's delayed because it is monitoring what these parallel systems are doing. And it's normative. It sets the standards by which you optimize your real-time control. So that's where the definition of consciousness came from. But let's now see whether this makes sense when we look at the brain. So we built a system called BrainCube. And actually, I built it. I can show it to you. We built it because I can never remember what all these structures in the brain really represent and what they do. And this is a very simple model of the human brain. And we can sort of zoom into it. It has 1,000 nodes, right? And the connections here are all calibrated in functional and structural terms. It's based on what's called the Heckman data set, right? So here we have a human brain that I can now manipulate. I can rotate it around and I can inspect it. And also I can ask it, okay, what do we really know about different areas? Because I can guarantee to you that even the most professional neurologist will not know by heart what all these nodes mean, where they're located, to whom they talk, and so on. So we build a system that allows you to do that. So actually you can click on a single node, you can see what it connects to, and also here you can then actually see what is our dominant understanding of that specific structure. You can also simulate it, you can lesion it, and one thing you can do with it is to try to understand how the human brain responds to distinct stimuli that we can measure with electrodes that are implanted in the brains of epileptic patients. Okay, so here you see the electrode placement of a single patient. The red dots indicate the distinct contacts that this electrode makes, and then we can do different kinds of, let's say, network analysis, or we can also replay the data we record from our patients together with the stimulation protocol, so we can online investigate how this brain might or might not operate. And we can also simulate in parallel the parts of the brain we're not measuring from. So we can really have a global view on how this brain might operate. So this tool is what we used to try to understand the brain of these epilepsy patients, but in particular to try to understand whether we could find any kind of confirmation for this hypothesis of a virtualized sequential delayed memory system that could possibly play a role in monitoring and optimizing real-time control. So this was our fundamental question. Okay, BrainCube also exists as an immersive virtual reality space. That's another story. So we started to look at the perceptual end of things. So we used a paradigm called induced blindsight. I will explain it in a little bit. On 3 patients, here you see over the different patients where the electrodes were placed. As in the end, we end up really looking at the temporal lobe, the medial temporal lobe, or the hippocampus, here indicated in yellow. You might see the little banana here. And also we looked at occipital lobe, which is a more visual area. This protocol is well established and it's been used by many labs. And essentially, you present a stimulus. In this case, it's a little line segment in one of 8 positions. And you mask it after 33 milliseconds. So that means it cannot easily enter consciousness. You control the exact duration of this for the perceivability of the subjects. There's some variability there. Then there is a pause in our case. So there's a memory component in the task, after which the subject has to say whether they have to indicate the location of this line segment. So they have to indicate 1 of 8. And then they have to also indicate whether they actually saw it. So you push a button that's from 1 to 8 to say, okay, I think it's there, in this position. And then you declare whether you saw the stimulus or not. It's a very standard paradigm. And if you look at the performance of our subjects, it is completely standard compared to healthy subjects, right? So if you look at the real locations at which you present the stimuli and the locations where people report them, their probability to be right is very high. So people perform the task very well. So now the first thing we did is see, can we predict the location of the target by just looking at the response in the brain across all the electrodes. So if we just sum all these signals, okay, does then that signal amplitude itself predict, or possibly predict, where the stimulus was? And since we have 8 locations, the random choice would bring you to about 12% correct. And you see that up to about 100 milliseconds, you can improve that prediction to about 20%. You can add 8% to your prediction by just looking at the amplitude of the signal that you get from these brains. But after that, your predictive capability drops very rapidly, right? So this seems to suggest that if you pick up a signal early on in the visual part of the brain, you can from there predict where the stimulus was, but you cannot really predict what the subject will say, okay? This again is consistent with current literature. So like here we have an experiment by the Dehan group using MEG, where they also show they have good prediction, a good correlation with distinct MEG responses across occipital and parietal cortex early on in the trial. And the same here we see a recent paper by the Roelfsema group in Amsterdam, where he measures from V1 to V4 to the dorsolateral prefrontal cortex, where essentially he's making the point that by just looking at the amplitude of the signal that he captures there, he can predict the response of the monkey in the task. So again, it's a pure amplitude-based interpretation. However, there's a problem here that up to 1 second, you have predictive power, but after 1 second, the predictive power is gone. So from then on, it's very difficult to predict where the subject will actually place that target, what the response will be. And this then led to a quite complicated analysis and proposal by these authors about where this memory might reside. Actually, it's e-life, it's not science, but okay. And from there, they come up with this notion or a new hypothesis, what they call silent memory, right? That the content that might enter consciousness in a later stage is maintained in a structural memory that is not dynamic and therefore not measurable with MEG. And that's the contention that I would like to investigate. So these two theories fall within the realm of what is called the global neural workspace, that also Jean-Pierre Changeux talked about earlier this week. And the global workspace has the idea that you can think of the brain as a bunch of interconnected processors that exchange signals. Okay, and they exchange signals in order to enter, let's say, a shared information space, a global workspace. And it's that the content of this global workspace that would then also define the content of consciousness. And a distinct signature of these processors to enter the global workspace is what's called ignition. A transient in the response, so an increase in the response, in the amplitude, that would indicate that that processor is now sending information into the global workspace. That means in this conceptualization of the global workspace, there's a strong parallel with what's called drift diffusion models of decision making. Every node is integrating its information to some threshold, and once it reaches the threshold, it passes information on. That's ignition, right? And these models have been used very extensively in the study of the neural substrate of decision making, and people have been very happy to see that the responses that you can measure in frontal cortical areas seem to reflect very directly this idea of the integration of evidence to threshold. However, what we have reported a few years back is if you make the task a little bit more complicated, let's say you add a memory component to the task, or you add a simple rule, this kind of integration models break down very quickly, and they lose all their predictive power. Okay? So this raises the question whether these kinds of signatures of ignition are possible to extract because the tasks themselves are relatively limited. And that would also explain why they are often occurring so close to the periphery, either the sensory periphery or the motor periphery. I mean, dorsolateral prefrontal cortex is one step away from the frontal eye field that in this task would control the action of the monkey. So this is something we have to, is the language, if you want, of the global workspace really expressed in the firing rate of neurons, is now the question. So then the second thing is, where's the memory that was not detected earlier? Is this memory a silent memory, or is there a dynamic memory that you might expect? And last, another problem is that rate-based systems don't scale very well. This is a big problem right now in, let's say, deep learning systems. Right? If you add a new stimulus, let's say a new class of stimuli, so far you'd have been detecting bottles really great, and now you want to do computers. The synapses, the weights in your network that you have dedicated to bottle detection, you cannot touch anymore because you will create mixture states. So you have to add new connections. So the scaling properties in that sense of these weights in the network are exponential, which is not very favorable. And moreover, it's not consistent with what we know about cortical connectivity. Right? Henry Kennedy and others have been showing for a while now that if you look at the wiring in the cortex, the majority of synapses remain local to the volume. So they don't show the wiring patterns that you might expect from a global workspace system or an integrated hierarchical system. We actually had anticipated this a while back already, introducing the temporal population code, which is very close to the ideas of John Lissman, who unfortunately passed away last year. John was really one of the great theoreticians of systems neuroscience. And John had advanced already in the 90s this concept of theta gamma coding, right? That the coding of the brain doesn't play out in rate terms only, how active am I, but more in terms of how the different frequencies at which neurons are responding relate to each other, which of course would also give you a higher dimensional representational substrate. And that's also the direction that we have taken. And that's also the hypothesis that we're going to apply now to answer these questions. All right, so we have problems to solve. What's the code? Where's the memory? And how global is the global workspace really? Is it global, like the whole brain, the whole neocortex, or should we include other structures, or should we limit it to subregions? Let's say frontal parietal networks. So if you want to look for memory, of course, an obvious point to start is the hippocampus, which we know is a structure deeply involved in episodic memory. And also we know a lot about the hippocampus, certainly from the fantastic work going on in neuroscience. It's really one of the most, I think, advanced areas within neuroscience right now. However, if we just look at, again, the amplitude of our recordings in either the visual cortex or the hippocampus, we see, if we have classifiers, if I have a classifier that is just reading the signal that I measure, and from there tries to predict where the stimulus is, I can have pretty good prediction when I just take the electrodes here, the occipital areas. I can predict really well where the stimulus was. But if I look in the medial temporal lobe, so the hippocampus, if I take my contacts in this region, I have no predictive power. So this shows you that if I move up this processing hierarchy in the brain, rate as such is not informative anymore, at least not for these electrodes that we look at. So, moreover, if I just compare the responses I can measure in the medial temporal lobe for the seen correct trials, where the subject knew what they were doing, and to compare those to the unseen incorrect trials, I don't see any difference. Right? So I cannot recover the location of the stimulus, so I cannot say anything about the sensory state, and I also cannot make predictions about the response state. However, if we now shift up one gear in our coding regime and we start to look at frequency, the game changes rather dramatically. So here we are looking at all the trials where the subject correctly indicated the location of the stimulus and also declared that it was seen. And we compare it here to those where it was not seen and the choice was incorrect. And this is frequency that we measure. And we see a marked increase in the power, so the presence in the signal, of low frequencies in the theta range. Okay? So apparently, even though I cannot recover this in the amplitude, something is happening here in the frequency domain, so we should try to figure out what this is doing is this a signal that is sort of coordinating this dynamic memory now? So is this the memory that De Haan and his crew could not find? So let's take a look at that. And for that, we go to the phase amplitude coupling. And in that case, basically what you do is rather straight. You just say, look, I have a bunch of oscillators, and some are active at low frequencies, and others are active at high frequencies. And all you try to measure now, or quantify, is to what extent the phase of the low frequency, so when they go up and down at a low frequency, to what extent they change then the amplitude of the high frequency. So is there a coupling between the two? Which, when we talk about theta gamma coding, this will be clearly expressed. And this is also a measure, of course, widely used in neuroscience by people like Pascal Fries or Earl Miller, but they often interpret it mainly in terms of how structures in the brain communicate with each other. And here we try to see if it's really also a representational substrate. So what we see now is if I just take my electrodes in the medial temporal lobe, so we just measure now from the hippocampus, and I try to look at all these possible couplings of frequencies and how they modulate each other. So if I compress that data here, we can look at this plot. We see if we go from the start of the trial over our 3 subjects, here comes the stimulus, here's the memory, here's the retrieval of position, here's the retrieval of confidence. You see that over this whole collection of coupled oscillators that we now just try to capture in a single number, there's a very distinct pattern that very accurately can predict in what phase of the experiment I'm in, including the memory and the retrieval of the 2 targets, right? So this suggests now that the memory structure underlying these tasks, that like in induced blindside, capitalize not on the firing rates of memory systems, but they capitalize on the distinct dynamics that they maintain across each other as expressed in phase amplitude coupling. So now we're going to unpack this a bit further. Okay, but how is this now a memory? Right? How can you have information in that system? Maybe it is just a non-specific expression of a dynamical state that has nothing to do with remembering a location in an induced blindside task. So for that, we made the task a bit more complicated. And now the subjects are navigating in a virtual reality environment. And given specific locations they're at, they get presented with a stimulus that they will be asked to recall later on. Okay? From an episodic memory perspective, you can see that we control the context, the environment you're in, their global landmarks, the towers, and so on. And then there's a local item, like here the envelope. All right? So we're building episodic memories, and the question is, how are these episodic memories now encoded, and how could it possibly explain the maintenance of the memory that would serve conscious detection of stimuli? So we have 10 patients now. We look at anterior hippocampus, a single contact indicated here in red. So let's move forward. First, we do 2 things. First, look at what's the impact of volition, and then we look at what's the structure of the memory. Here we have 1 patient navigating the environment. Here we have the real-time record coming from its frontal cortical electrodes, parietal cortex, anterior hippocampus. And here we plot the correlation, the mean correlation over bins of 100 milliseconds. And here we just have the binary version of that for the positive correlations to show the global structure of how different brain areas interconnect in such a trial. But now, does it make a difference when you have volition or whether you are yoked to the path of another subject, right? So we use a conditional entropy measure to just get a feeling, to quantify the structure of the network that we're reading out across our electrodes. And this is a conditional entropy measure that allows us to look at how these networks change over time. And the main thing that matters right now is if we take a random network, that means we take the same nodes, but we randomly change the statistical structure that we have measured from our patients. So that's like the baseline. We are strongly deviating at important points from a random network. So there's structure in this system. So what's that structure exactly? Or is an active structure different from a passive structure? That's the case. So active is red and green is the passive subject. So that would suggest that across all these electrodes that allow us to have a snapshot of the underlying neural network of the cortex and the hippocampus, the way this network is dynamically reconfiguring itself continuously, dependent on these task conditions. Okay? And what we see on top of that is again a strong modulation of the theta frequency in the hippocampus in the active condition versus the passive condition. And now I want to understand what is theta modulation that is tied to this active condition, what role it plays in the formation and the readout of memory. So here we go to 7 more patients. In red, you see the electrodes we have in the hippocampus. In blue, we have the electrodes in the cortex of the lateral temporal lobe. So we can start to compare how cortical responses and hippocampal responses relate. And again, the subjects are in a maze, so now they have to detect stimuli or remember stimuli in certain locations, so we can ask, well, how does the memory now respond when the location in which you had to remember something is consistent with when you stored that information or inconsistent, so congruent or incongruent. We use reinstatement analysis, which is a very beautiful way to abstract complex data of the brain. So basically what you do, again, it's really simple. You have your signal, you look at the power spectra over time, so you have a bunch of frequencies popping up, they have power, they have presence, and you just plot it over time. That's in the plot that we have here. And then you compress that in a single vector. So you say, okay, so this template of frequencies is apparently representing the state of the network. So then, of course, I can say, okay, if I now recall the memory, do I see that same template? And when do I see that in time? Right? So it's a way to compress this complex data, but purely in terms of the frequency response. And then you can look at the similarity map, like, okay, how well do then these frequency templates align when I encoded the information or when I retrieved that information? So what do we see? During encoding, in the same location, so you bring subjects to, again, the same room in the virtual maze, you present them with the stimulus they have seen before or they have not seen before, and you ask them, do you recognize this? Okay? And now what we see is that if there's congruence, you see the same stimulus in the same room, that these frequency templates that we use to encode the storage of this information reappear. There's a very strong correlation. But you see that during the storage of the information, during encoding, these templates were expressed from 0 to about 3 seconds. But in the recall, they all get expressed in 1 second. So you see there's a very strong time warping. Right? So the hippocampus is playing back this information, advancing it in time. So it's not following analog time, which is an important challenge again to rate-coded systems. How do you want to time warp if you depend on analog codes? If you look now at the difference between incongruence, so the context effect, you see that's a very distinct difference, which also shows this time warping, right? So you see a very strong time warping that plays a role in these congruent spaces that is not present when the location is not the same in the recall. And this effect is highly significant. So we can recall in a more efficient way when we are in a congruent state, but you also see we are time warping, we are time shifting our representations. If we now look at the cortex, the lateral temporal lobe, that here we see, if I just look at the items, I compare my responses to the items during acquisition and during expression, there's a high correlation throughout in the cortex that you don't find in the hippocampus. While if you compare different items with each other, you don't see that strong correlation. So again, a single item, the envelope, the bird, the guitar, has a very unique fingerprint in this power spectrum. Okay? And here the difference is again expressed. But now what we asked, because we want to bring it to the contribution to consciousness, is okay, how do these responses change if you ask the subject about their confidence of their rating? So now that they say, I've seen this before, and I'm very confident about my judgment. So what we see here is that there's a very distinct coupling going on between these encoding templates and the retrieval templates that is uniquely expressed when you're highly confident about an item, but this confidence is expressed in the lateral temporal lobe, so in the cortex, not in the hippocampus. So this would suggest that the hippocampus is sort of organizing and maintaining this dynamic memory state, but the readout and the translation to the conscious state depends more on a cortical process. So we have also then further shown this very distinct coupling between hippocampus and lateral temporal lobe. So we have some answers, right? So on the one hand, when we talked about the code, there was this debate, is it rate, is it ignition, is it something else? And I see more a mixed mode operation where close to the periphery, this notion of an ignition, of an analog change in the amplitude is relevant. And we saw we can recover stimulus properties with that. But in terms of the internal space, the internal mental space in which we operate, the coding regime is critically different. It has nothing to do with rates anymore for a number of reasons. One of them is, by making my encoding substrate frequency dependent, I can actually morph it in time, okay? Otherwise, that would be very difficult. So where's this memory? Well, I think I've shown to you that it's safe to believe that also in induced blindness, the memory is not silent and structural, it is dynamic and it depends on the temporal lobe, as we might have expected. Why did people not report? Well, they use scalp electrodes, or they never looked for it. Okay, but it's not such a crazy idea to believe that memory plays an important, or memory depends on hippocampus. All right, so we jump over this one. So we entered the room with the theory of consciousness and a distinct definition, which very much emphasized this notion of virtualizing the world in order to extract value. Okay? Have we explained that full story? No. But I think we made a few steps forward, because what we clearly saw in this hippocampal memory, it is definitely virtualizing. It's not analog memory, right? It's independent of analog states. So right now we're looking at the interlinking to the valuation systems and how this then gets expressed to optimizing the real-time control. And for that answer, this is work that's going on right now. But what you see again is that the robot experiments have actually helped us to rethink in functional terms what consciousness was good for, could be good for, but in a way that we could make it testable in real physiological experiments with humans. And now we're also bringing it back to the robot experiments to understand this question of valuation better than we did before. Thank you very much.
Thank you very much, Paul. We're running a little late. We can take one or two questions while we set up for the next talk. Over there.
Yes. Working memory. We've got theta, and theta between the hippocampus and frontal, but I think especially anterior medial frontal. Mm-hmm. And then the gamma is holding the individual themes, the individual
items.
Content,
Yeah.
items, right?
Yeah.
But the theta seems to be what's maintaining the
What coordinates
working memories.
them.
Is that basically correct?
Yeah, that's so. What is amazing, like John came up with this idea of theta gamma coding in the 90s, and it never really sort of got a lot of traction. But that's really what we see here, right? That you have these sort of lower frequencies as coordinators of a broader memory network that are playing out in much higher frequencies. So this is really the relation that we show here. Absolutely right. Yeah.
Fantastic.
Hi, great talk.
Thank you.
My question's pretty quick. I'm just curious how someone like me, an undergraduate student, could possibly get access to the BrainCube software just for research purposes.
Write me an email and you get it.
Cool.
Thank you. Sure.
All right, let's give Paul a round of applause for a great talk. Thank you very much. Okay, our next speaker is Selen Atasoy, now at the University of Oxford, Department of Psychiatry. He'll speak on Connectome harmonic signatures of consciousness and its altered states. Selen.
Thank you, Stuart, for the introduction, and also would like to thank you and all the organizers for the invitation. It's really a delight to be here again. So I'll speak to you today about the Connectome harmonic signatures of consciousness and its altered states. And with these altered states, I specifically mean the psychedelic states that we heard all these fascinating talks about on Tuesday, then meditation, as well as sleep. Now, in all these states of consciousness, there is a change in our conscious experience, obviously, but mostly in the psychedelic state, and I would argue also to a large extent in meditation, and maybe even to some degree in the sleep state, in dreaming or early stages of sleep, there can be a certain type of awareness of our own unconscious thoughts. So it really seems like we are in a way diving into the depths of the unconscious. So I'd like to start with this question. What happens when we dive from the open waters of consciousness into the depths of the unconscious? And if our attempt is rather successful. Just a quick note, actually, the elephant was apparently playing in this picture. So today, we really can approach this question from rather a scientific point of view using functional neuroimaging data such as fMRI, MEG, and EEG. And we also heard about yesterday, there's, over the last decades now, there's an increasing interest in neuroscience community in the neural correlates of consciousness, but also recently on its altered states, on the neural correlates of psychedelic states, meditation, as well as sleep. So what I'm going to propose today is that, that we slightly reformulate this question from what are the neural correlates of consciousness to what kind of music is your brain playing in consciousness and in its altered states. And please bear with me, even if it doesn't make sense to you yet, and I will try to demonstrate that actually this reformulation may help us extract the characteristic signatures of these states. But before we do dive into the depths of the unconscious and look for the harmonic signatures or Connectome harmonic signatures, I wanna show you a really nice illustration of harmonics and their self-organization in this video. Actually, this video is a very nice evolution of a very old experiment first performed by a musician called Ernst Chladni in the 18th century. And you probably have noticed this complex sand patterns forming on the metal plate when he was playing a musical note on the keyboard. Now, this is happening because any system, even a simple metal plate, has certain preferred frequencies called its natural frequencies. And if you excite the system with one of these frequencies, then it will resonate and it will vibrate. And this vibration is going to induce a standing wave on the metal plate. Now, when the standing wave emerges, that sets up a pattern of oscillation. And as we can see, for instance, in this slide, the red parts of the metal plate move down and the green parts will move up, and they will swap. And of course, the sand is going to be thrown off from the parts which are moving the most, and it will accumulate at the boundaries of these regions, which are the still points, forming these complex sand patterns. And in fact, this is how we create music. For every single musical note that we play in the musical instrument, there is an accompanying standing wave pattern emerging in it. And even though these patterns are quite complex, or they look quite complex, and very different from each other, we can determine their precise shape by solving one simple line of equation. And that equation is the eigendecomposition of the Laplace operator applied to the shape of to the domain of the metal plate. Now, what this equation tells us is it gives a spatial part which describes the shape of the standing wave, which is the harmonic pattern, and it also gives us the eigenvalue, which relates to the temporal aspect of it, which relates to the natural frequency of vibration. And what strikes me the most about this equation is that nature seems to be using it a lot. So it not only describes these, the shape of the standing waves in metal plates or musical instruments, but it also describes the electron orbits and atomic energy levels in quantum mechanics, or electromagnetic interaction patterns within a grid of ions. And probably most strikingly, it gives us the building blocks of biological pattern formation and morphogenesis. So these complex patterns that we observe on animal coats are actually a combination of this type of harmonic waves. And in fact, if we were to cut the metal plate in shape of an animal skin, like Murray and his colleagues did in their very interesting experiment, we would observe that the standing waves emerging on these particular domains would resemble different types of animal coat patterns for different frequencies. So the thing to notice here is that actually the equation that I showed you, the eigendecomposition of the Laplacian, links in a way space, time, and geometry, in this case for the particular example of mechanical vibrations. Now let's consider for a moment the puzzle we are trying to solve about the brain. Here again, we have certain types of dynamics, in this case caused by neurophysiology, by the interaction between excitation and inhibition acting on neural populations, which then gives rise to brain's resting state networks, so networks of distributed brain regions that exhibit spontaneously synchronized activity over time, as we have now discovered over the last 2 decades. And it has long been known that brain activity oscillates over time, and these oscillations cover a very wide range of frequencies, ranging from 0.05 Hertz to 500 Hertz. And finally, we also know that both the temporal oscillations as well as the resting state networks are actually constrained and implicitly linked to brain's anatomy. But today we do have the technology to create a detailed mapping of human brain anatomical connectivity. So we can trace the long-distance white matter thalamocortical fibers connecting different parts of the human brain, and we term it, the set of all of these connections, as the human connectome. So we know that this human connectome actually constrains the neurodynamics. So these parallels actually led us to hypothesize, what if the same fundamental principle that explains various different physical and biological phenomena could also give us an explanation or a link between brain's oscillations, resting state networks, and its anatomy. Now, in order to test this hypothesis, in our 2016 study, we did solve the very same equation, the eigendecomposition of the Laplacian, this time applied to the particular connectivity of the human brain, to the human connectome, which included both the gray matter connectivity as well as the white matter connectivity. And as you see here, the spatial component of this equation yields these widespread distributed patterns of correlation that we call connectome harmonics. And as expected, the complexity of these patterns increase with increasing frequency. So for increasing frequency, we get more complex and more distributed patterns. But what came as a nice surprise in that study was that the resting state networks reported in the literature have actually significantly matched a different part of the connectome harmonic spectrum, especially in the first 40 to 50 low-frequency connectome harmonics. So in a way, each of them had their own unique connectome harmonic signatures. And this discovery then led us to hypothesize, what if brain activity in different states of consciousness also had different or unique connectome harmonic signatures in a way? And in order to test this, we decomposed brain activity measured by functional magnetic resonance imaging data into the activation of connectome harmonics. So for each time instance, we take the cortical activity pattern measured by fMRI, and then we decompose it into the activation of different connectome harmonics. So in a way, we estimate the contribution of each of these connectome harmonics in order to create this particular pattern of cortical activity. And based on that, we can then compute the power and the energy of each of these connectome harmonics. So the power would tell us how strongly that particular connectome harmonic contributes to brain activity, and energy energy in a way combines this power with the intrinsic energy of that particular connectome harmonic. So you can think of it as a frequency-weighted version of the power. So if we recall the harmonic patterns emerging in musical instruments and in metal plates, what we are doing with this decomposition is actually not very different, or actually maybe not different at all, from taking a complex musical piece and decomposing it into its musical notes. Now, using this technique, I would like to now show you what kind of music your brain is playing in the psychedelic state induced by LSD. So to understand that, together with Robin Carhart-Harris and his group, we have applied the connectome harmonic decomposition to the fMRI data of 12 healthy participants who received either a dose of LSD or a placebo. And the scanning happened in actually 3 different sessions, so 3 scans. In the first and third scan, the participants were simply resting in the scanner, eyes closed, and in the second scan, they were also listening to music. So what we found quite interestingly, but maybe not that surprisingly, was that LSD led to an increase in the total power and the total energy of brain activity when we considered all the connectome harmonics all together. And if we look at the probability distribution of observing a certain energy level, we saw a clear shift towards high energies under the effect of LSD, meaning that it was way more likely to achieve these high energy states under the effect of LSD. So next, we wanted to know which of these connectome harmonics caused this type of energy increase that we observe in brain activity. So here you can see the energy for all 3 of the scans, we see the energy of the whole spectrum of the connectome harmonics indicated by their wave number. And if you look more closely and look at the difference between the LSD and the placebo condition, we see that there is a clear suppression of the low-frequency connectome harmonics and a significant increase in the energy of high-frequency range. Now, it's probably important to note that the x-axis is shown in logarithmic coordinates, so it's really a narrow range of low-frequency harmonics that are suppressed, which also coincide with the range where we found the resting state networks. And a very broad range of connectome harmonics increased their activity. So next, we wanted to know whether these type of energy changes that we observe in the brain had any meaning for the subjective experience, for the subjects themselves. So we looked at the correlation between these energy changes and the intensity of certain subjective experiences that the subjects have reported. So firstly, we found that the suppression in the low frequency range actually significantly correlated with the intensity of the ego dissolution, with the experience of feeling no boundaries or being one with the universe. And quite interestingly, the very same range, the suppression of the exact same range also significantly correlated with the intensity of the emotional arousal. However, that suppression was not enough to account for the intensity of the positive mood. So the intensity of the positive mood actually correlated with a broader range of frequencies, with the energy changes in a broader range of connectome harmonics, which contained both the suppression of low frequencies as well as the increase of a slightly higher frequency range. Now, these results are kind of encouraging and in a way indicating that it is not only that we do find some changes in the brain, but they do have a meaning or an equivalent in the experience of the subjects. So as next, we want to explore the repertoire of the connectome harmonics without distinguishing between different frequencies. So we looked at the probability of observing a certain amount of activation. So here what you see in this plot is the tails of the plot would tell us what is the probability of any connectome harmonic playing out really loudly. And the 0 here would indicate what is the probability of any connectome harmonic being silent at any given time in the fMRI scan. And what we found for the LSD case is that this probability of connectome harmonics being silent actually reduced, it decreased, which then means that actually LSD expands the repertoire of active connectome harmonics. So in the musical analogy, that means that your brain is actually playing more of these musical notes simultaneously when it's affected by LSD. And that's quite interesting because there are actually studies which report in the literature that improvising jazz musicians play significantly more musical notes during improvisation compared to memorized plays. So that seems to really, in a nice way, parallel our findings on LSD, what LSD does to your brain. It activates more of the connectome harmonics simultaneously. But then, of course, a question that naturally follows is, well, maybe it is randomly activating the connectome harmonics like a child pressing more of these, the keys of a piano simultaneously. But it turns out that this repertoire expansion is actually happening in a non-random fashion. So when we look at the correlation across different frequencies in terms of their activation over time, we found quite a bit of increase in a very broad range of high-frequency connectome harmonics, which actually coincides also with the range which increased its energy. So this actually indicates that there is a spontaneous but structured reorganization occurring under LSD rather than a random and chaotic activation of the connectome harmonics. So your brain is really not randomly playing different musical notes, but it seems to be playing rather more like an improvising jazz musician with a complex and spontaneous order. Now, there is actually one particular situation when dynamical systems show this type of increased repertoire and a complex order in their behavior or activity. And that is when they actually approach what is called criticality. So criticality is this special type of dynamics or special type of behavior that occurs just at the transition between order and chaos, just at the border of these two different opposite type of traits where these opposing dynamics are actually balanced. And to give you a little bit of an intuitive understanding of criticality maybe, it said criticality is the constantly shifting battle zone between stagnation and anarchy, the one place where a complex system can be spontaneous, adaptive, and alive. And it's where life has enough stability to sustain itself and enough creativity to deserve the name of life. So in a way, the dynamical system, or any dynamical system close enough to criticality, has enough stability to function in a meaningful way, but also has enough freedom to improvise and to create something rather novel. And there is one particular characteristic signature of criticality that any system would show, that you can search for, and that is the emergence of the power laws. So if you were to plot two different observables, two different variables in the dynamical system, if it's at criticality or close enough, then they would follow power laws, meaning in logarithmic coordinates, they would have a linear relationship. And it turns out that we do find this type of power law characteristics in the power-frequency relations in fMRI data when we look at the data with the Connectome harmonic decomposition. So here you can see for all three different scanning sessions, the power versus wave number plotted across the Connectome harmonic spectrum. In the top, you see the maximum power, in the middle, the average power, and in the bottom, the power fluctuations. And it's quite clear that all these relations follow power laws for both cases, actually, for placebo as well as LSD. So that means that resting brain is already close enough to criticality that we observe certain signatures. But we did find quite remarkable differences when we looked more closely for the LSD versus the placebo case. And that was, first of all, we observed a decrease in the power law exponent. So here you see these bars decreasing, which tells us that actually the slope of this curve of the power law is actually decreasing under the effect of LSD. So it's really actually affecting the power law characteristics. But furthermore, we observed that the error of fitting the power law, so how closely these data points actually follow the line, how closely they follow the power law, that also changed under LSD. The error decreased under LSD, suggesting that there is actually a tuning of brain dynamics further towards criticality, because we observe, in a way, more enhanced signatures of criticality under LSD. So let me briefly recall what I have showed you so far in terms of the Connectome harmonic signatures of LSD. First, we observed a significant increase in the total power and the total energy of all Connectome harmonics under LSD compared to placebo, and there was a higher likelihood of reaching higher energy states. And then we also found that it altered brain activity, LSD altered brain activity in a frequency selective manner. So it suppressed the energy of low frequency range and increased the energy of a very broad range of high frequency Connectome harmonics. And we also found rather more enhanced signatures of criticality in the power laws, which indicated that there is a tuning of brain activity rather towards criticality happening under LSD. Now, are these changes specific to LSD, or would other psychedelics also lead to similar Connectome harmonic signatures in the brain? So in order to see that, we did also look at the effects of psilocybin in the brain, which is still in preparation. So I'm going to be showing you from now on a little preliminary results in different states of consciousness. So we looked at the fMRI data of 12 healthy participants under the effect of psilocybin, which is the psychedelic compound found in magic mushrooms, and compared it to placebo in eyes closed resting condition. And here again, we found a significant increase in the total power and the total energy of brain activity in the psychedelic state, in a psilocybin-induced state, and also a shift in the peak of this probability towards higher energy states, making them more likely to happen in brain activity. We observed again a similar type of suppression in the low-frequency Connectome harmonic range and an increase of energy in high-frequency range. And once again, we observed a change in the power law characteristics. So the power law exponent actually decreased under the effect of psilocybin, and the error of the power law fit also decreased, suggesting that there's a tuning of brain activity rather towards criticality. Okay, now moving from the psychedelic state to another quite interesting altered states of consciousness, to meditation. We looked at the fMRI data of 16 experienced meditators and 16 healthy controls during mindfulness meditation as well as during resting state. So to our surprise, we actually found a significant increase in the total power and the total energy of all Connectome harmonics considered together for experienced meditators during meditation, so which is the light blue bar here. So in a way, that means when you silence the mind, it actually increases the energy and the power of your brain activity, which is also quite evident in the probabilities of observing certain energy levels. So the light blue curve here peaks at very high energy values, which is the curve for the experienced meditators during meditation. And if you look at the dark blue curve, which is the healthy controls during meditation, we see this little bump here in that energy range, which means that there is a bit of an increase in the energy, in high energy states in their likelihood, but they don't seem to be able to shift their dominant energy state to that particular range. And if you look at the differences between the meditation and resting state, here again you can see the so the meditators are the blue bars, and the control group is shown in red bars. And very strikingly, we found a significant increase in the high frequency Connectome harmonic range, which coincides with the psychedelic experience, where we found the same energy increase. And this, we did observe the increase in energies for the control group as well, but it was way more significant for experienced meditators. We didn't observe that type of suppression of low frequencies as much as in the psychedelic case, but when we look at the signatures of criticality, quite interestingly, we found again a decrease in the power law exponent, which is the slope of the power law, during meditation for experienced meditators. But maybe a curious finding is to point out is that we did observe a decrease in the error of fitting the power law to the data points for experienced meditators in the resting condition. So now there is a possibility that this really reflects the long-term effects of meditation in brain activity. But to be a little more skeptical, there is also, I mean, it's also highly likely to reflect the individual differences in the meditator group, because some of our experienced meditators had more than 10,000 hours of meditation, whereas some had only 2,000 hours. But if you look at the whole picture, there is actually a striking similarity between the Connectome harmonic signatures of the meditative state and the psychedelic state. So our results in a way actually suggest that you may be tripping on your own resources if you have enough experience with meditation. So finally, I would like to share with you some of our preliminary results from our sleep study, where we looked at the fMRI data of 18 healthy participants in the wakeful resting state, in non-REM sleep stage 1, which is light sleep, non-REM stage 2, and slow wave sleep, which is the deep sleep. Now, in this case, we have found that actually deep sleep, which is shown in the dark blue here, has the exact opposite effect of the psychedelic state and the meditation. So the total energy and the total power of brain activity actually decreases during deep sleep. But very interestingly, light sleep showed similar signatures to psychedelic and meditative states, such as increased total power and increased energy, and a higher likelihood of reaching the high energy states. And when we actually look at the difference between resting state, a wakeful resting state, and each of these sleep stages, we do see also a similarity between light sleep, which is the light bars, light blue bars here. We observe an increase in the energies in rather the higher frequency range, where we observe exact opposite effect for deep sleep. So if you recall that psychedelic state was actually suppressing low frequencies, we see an increase here in deep sleep. And again, in the opposite way, we see a suppression in the higher frequency range. And when we look at the power law characteristics, we again observed an enhanced power law, like enhanced signatures of criticality, a change in the power law exponent as well as in the fitting in light sleep. So light sleep showed rather enhanced signatures of criticality, if you like to interpret it that way. And deep sleep actually showed rather diminished signatures. So in a way, our findings suggest that there are unique signatures of unique Connectome harmonic signatures which are characteristic to each of these altered states of consciousness as well as the resting wakefulness. And before finishing, I really would like to emphasize that by using the Connectome harmonic framework, we really do nothing other than changing our representation to this harmonic representation, which then allows us to look at the power and energy of different frequency Connectome harmonics. So maybe after all, the genius of Nikola Tesla was rather underappreciated in our community. As he said, if you want to understand the secrets of the universe, think in terms of energy, frequency, and vibration. And with that, I'd like to thank all my colleagues and collaborators who made this work possible. Morten Kringelbach and Gustavo Deca for lots of valuable discussions. Anira Eskrich, who collected the meditation data. Robin Carhart-Harris, Leo Rosman, and Mendel Killen for generously sharing their rare and beautiful psychedelic data sets. And Joel Pearson and Isaac Donnelly for their contributions in the development of the Connectome harmonic framework. And would like to thank you all for your attention.
Great question.
Thank you so much. As both a physicist and a professional musician, I really appreciate you taking that time to make those connections. I was just wondering, what about the patterns of coherence and harmonics set up in the rest of the body in this process? Have you thought about that, and do you have any plans to study that?
Thank you. Awesome question, really. I mean, as neuroscientists, we rather tend to focus on the brain, but I really appreciate this question. We haven't looked at it, obviously, yet. But to focus more on the brain-body connection, I think it would be a very interesting way to dive into.
Thank you.
I just want to commend you on your work as a fellow neuroscientist. I know that it's not easy to compute these measures. It's a lot of work. It's a great job. For anybody who has seriously experimented with psychedelics, they would tell you that phenomenologically, there's a difference between LSD and psilocybin. I'm just wondering how these Connectome models that you presented to us could account for that difference.
Yeah, so thank you for the question again. If the method is going to be sensitive enough to pick up on the differences between different, between psychedelic state induced by different types of psychedelics, we need to explore in more detail. So the psilocybin study is really preliminary yet. But I don't know if you were able to see it. I didn't comment on that during the presentation. There's a little bit of a shift in the suppressed frequencies and a change in the curve of the suppression. So we need to look more in detail into these kind of characteristics and maybe the co-activation patterns and so on to see if the method is going to be sensitive enough to pick up on the differences. I can maybe share of some really preliminary results on our ayahuasca study, which is going on at the moment, where we do see very different effects, actually, rather in between deep sleep and LSD-induced psychedelic state in ayahuasca, for example. But that needs to be explored more in detail.
Hi, great talk.
Thank you.
Can you also try and use this method not on a resting state fMRI signal, but task-related? So, for example, to retrieve a picture or a navigation to your house, or just think about yourself and then to check the Connectome harmonics. Can you try to do it also? Did you do it?
You mean task-based fMRI?
Yeah.
Yeah, we did discuss that a little bit. I mean, technically, there is nothing that says that can't be done. So, because we just take the fMRI and we decompose or we change the representation. So, technically, it is possible, but we haven't looked at the task-based fMRI yet, but that's hopefully coming soon.
Thank you for the wonderful presentation. Do you think that we may be able to point to specific connectome harmonics and isolate any functional or phenomenological significance? And likewise, do you feel that pathological states, such as those in the DSM-5, might leave semi-unique fingerprints on connectome harmonic states?
So, in terms of functional, like the first part of your question, in our first study, in the 2016 study, the reason that we got really excited about this work was the fact that we did observe a significant match between the default mode network and one of the harmonics. So it really suggests that there is kind of a relationship between functional networks of the human brain and this type of harmonic patterns. We are exploring that further in the moment with larger sample sizes and so on. And in terms of the pathological cases or psychiatric disorders, so we haven't started it yet, but in the next few months we are going to be looking at data sets with PTSD and depression to see if we can extract any type of connectome harmonic signatures in those states.
All right, thanks for the talk. So I have a question on the underlying assumption of connectome, or the assumptions underlying the idea of connectome harmonics, which is, so you made the analogy from vibrating plates to the skins of animals to the brain. In the case of a vibrating plate, you solve the Laplace equation and you have a complete set of orthogonal functions that could represent any function on that surface.
Exactly, yeah.
But there's an underlying physical mechanism that makes that useful. I mean, it seems to me, just to make a pathological example, you could take a map of the surface of the Earth and decompose it into spherical harmonics. But
absolutely.
Would that tell you anything? That's my question.
Yeah. So because I wanted to share the signatures of different states, I had to skip that part of the talk. But 2 years ago, I had shared actually our modeling results as well, which show how these harmonic waves or harmonic patterns emerge from the interaction between excitation and inhibition. So there is rather a biological link to it that we also further explore, but if you're further interested, we can either talk offline, or I would also refer to the study.
Okay, thank you.
Really interesting talk and beautifully presented. I wanted to ask about dose. As you were talking, I was wondering about dose and the parameter of order, chaos, and criticality, how that would play with true microdosing, below 20 micrograms, maybe the museum dose, so-called standard dose, and then the heroic dose. Does it really change if you get into those different levels of exposure?
I think it's a great idea. So I mean, I would suspect that they would affect the critical dynamics. Microdosing, like looking at microdosing LSD data sets would be a great study. Maybe we should talk with Robin to collect the data for us.
Yes, so your x-axis was wave number. can you convert that into hertz, temporal frequency, or?
Well, I really rather shy away from that because the only thing that we know really for sure with the Connectome harmonics is their order. So we have not related the spatial frequency or spatial wavelength to the temporal oscillation in terms of hertz yet. That could be maybe done by modeling studies or by looking at matches of the harmonics in MEG data, but we haven't done either, so I would rather not give you any range yet. But,
and yeah. would your results be then, if not qualitatively consistent with direct EEG measurements, and you take a spectra of EEG activity and look at the power in different frequencies?
Well, we haven't looked at any EEG data set yet.
But it should turn out to be consistent at some level with EEG data.
The conclusions are rather consistent so far. For example, what Anil has presented 2 days ago, they are looking at LSD MEG data sets, and they find very similar results, or like an expansion of the repertoire of states and very matching results which are in line with what we find in fMRI using a very different technique. But yeah, we haven't looked at MEG or EEG data set yet.
Okay, thanks.
Next question. Thank you very much for your inspiring talk. I was wondering, do you think there could be any kind of connection between these Connectome harmonics and the geometric nature of visual hallucinations?
Sorry, can you repeat the question again?
Do you think there could be any kind of connection between the harmonics you're talking about and the geometric nature of visual hallucinations?
Geometric nature, yes, there is actually. So there are quite a lot of modeling studies and a huge literature to be mentioned about the hallucinations, which use very similar type of computational models that we have used in our 2016 study. And they show that actually hallucinations are rather a combination of a few harmonics, or sometimes even one, but emerging on the visual cortex. So if you take into account the retinotopic mapping from the visual field to the visual cortex, and you just take the harmonics and map them, you would get the types of hallucinations that people experience. So there is definitely a quite strong relationship between the two. Yeah.
So I wanted to ask that, have you ever studied meditation versus deep thinking, or when children are engrossed in some focused activity, how the fMRI looks like?
We haven't looked at it yet. So in this data set that I presented for meditation, the naive subjects were instructed to focus on their breathing, but that was rather the first stage of the mindfulness meditation, not a focused meditation. But if anybody has the data set, I'm very happy to look at it.
All right, let's thank Selen and Paul for a great session.